This method can help understand key questions in the immune cell migration field, such as how the cytoskeleton sends shear forces and how integrin ligands and chemokines affect flow-induced migration of leukocytes.
The main advantage of this technique is that it's a new type of migration assay that's easy for beginners to perform. It uses only commercially available equipment and reagents, as well as free software.
Though this method can provide insight into marginal zone B cells, it can also be applied to other immune cells, such as activated T cells.
On the day before the experiment, thaw an aliquot of ICAM-1 and dilute it in PBS to a final concentration of 5 micrograms per milliliter. Then, add 30 microliters of ICAM-1 to the desired chambers of a flow slide. Place the slide into a humid chamber and set it at 4 degrees Celsius to incubate overnight. The next day, wash the chamber by adding 100 microliters of PBS to one well and then withdrawing 100 microliters from the other well of the chamber. Then, add 100 microliters of blocking buffer to a chamber and incubate the slide in a humid chamber at room temperature for 90 minutes. Next, wash the chamber by adding 100 microliters of PBS to one well, withdrawing it from the other well, and then add 100 microliters of migration buffer to the chamber. The slide is now ready to use. Store it in a humid chamber at room temperature until the experiment. First, plug in a fluid pump and attach the fluidics unit. Then, turn on the pump software.